Good evening. Welcome to today's session. This is the sixth live practice session for the NPTEL course Environmental Biotechnology, and I am Guru Priya, Prime Minister's Research Fellow, doing my doctoral research in the Environmental Engineering Division at the Department of Civil Engineering, IIT Madras. So every week, based on that week's lecture content, we are solving some practice questions. So the same we would do this week also. If there is any issue with the audio or the presentation of the screen, please let me know in the chat box. And before starting, if you have any doubts from the previous sessions, please let me know in the chat box. I'll wait for a couple of minutes. And in today's session also, if you have any doubts in between, you are free to ask in between the sessions. Okay, so can I get a thumbs up if my audio and the presentation is clear? Can you please let me know if you can see a screen being presented by me, and also if the audio is clear? Okay, so like the previous sessions, we will go with solving some question and answers only. So I'll present the first question for today. For like the previous weeks, for each questions, I'll give a few minutes for you to solve and post the answers in the chat box, and then we'll discuss the answer for the question. So first question is slightly a lengthy question. Uh, you have to justify the following statements. If you feel the statement is false, please write the statement is false. Otherwise, like tell um, the reason behind the statement or the implication because of the phenomenon mentioned in the statement. If there is any doubt as in what you have to do, kindly let me know in the chat box. Okay, so here first you identify if the statements are true. Once that is done, the question is to justify the following statements. Like if only 0.01 to 1% of the total bacteria in the environmental samples are cultivable. If the statement is true, you have to tell why. This is just to give a general revision of the contents. And only one question will be of this type. Others are like the typical assertion and reasoning and match the following kind of questions that will be following after this. Okay, so we'll just see the answer for this question. So first of all the statements were right only. So the first statement is only 0.01 to 1% of total bacteria in the environmental samples are cultivable. So cultivable in the sense how much we can culture, isolate in laboratory conditions. We can only cultivate 0.01 to 1% because the majority of the environmental microorganisms have specific growth requirements in terms of nutrients and in terms of environmental conditions. So replicating those specific combinations of chemical as well as physical growth factors is very difficult in laboratory settings. So that is why only 0.01 to 1% of total bacteria in the environment are cultivable. So the second statement is 16S ribosomal RNA gene can be used as a phylogenetic marker for microbial identification. That is also true. We can use 16S rRNA gene because it is a structural component of the ribosomal subunit, 30S ribosomal subunit. And it is highly conserved across all bacteria. Conserved in the sense it is present in almost all bacteria, and there are regions in that which are similar across all the bacteria, and it also has regions which are different in different bacteria. So using the conserved regions we can identify the gene, and using the variable regions we can classify the organisms into similar types or similar groups. So in that case 16S rRNA is used as a phylogenetic marker for microbial identification. The third statement is direct microscopic counts using DAPI staining always exceeds viable plate counts. Can someone give what is DAPI staining in staining's full form? Okay. This is, DAPI doesn't stand for any technique name. It is a chemical name. It is diamidino phenylindole. Okay, so it is a fluorescent stain, and the stain can bind to double stranded DNA. So if we are going to do this microscopic counting of the number of cells using this uh, diamidino phenylindole stain, the number is always going to be more than viable plate counts. It is true because uh, some microorganisms, as we have discussed, are uncultivable. So if you are going to directly take the sample and do that, the DNA of that can be present. It may not be in a live form, but the DNA is present, and hence the fluorescent dye can bind to the DNA and still give reading. But when we are going to plate them, it is not going to grow. So in that case, the number of microorganisms identified using the staining technique is more than the number of organisms identified through plate technique. Okay, so the third statement is also right. And finally, coming to the fourth statement, environmental biotechnology processes are typically multi-organismic. So, environmental biotechnological processes use systems in which multiple organisms or microorganisms are involved, which is also true. The complex metabolic networks uh, involves like fermenters, primary fermenters, secondary fermenters, sulfate reducers, and methanogens in case of anaerobic processes. In case of aerobic processes also, we have studied about synergy, syntropy, and other mechanisms through which microorganisms exist in the ecosystem. Uh, so sometimes one organism depends on the presence of the other organism to maintain the equilibrium. And mostly environmental processes are involving multiple microorganisms, and it is not always talking about only one kind of microorganisms. So the first question was to identify whether the statement is right, and if it is right, you have to tell what was the reason. I don't know why most of you were not able to do that, but still, I hope it is clear now. If you still have any doubts, please let me know, and I'll now show the next question. So from the next question onwards, it will be like we did in the what we did in the previous sessions. So, for example, the next question, we have an assertion where it tells the activated sludge shows higher cultivability compared to other environmental samples. The reason is activated sludge contains organisms that are already adapted to controlled growth conditions. You have to tell. So I am not given the options here. I know I am like uh, clear that you would now know what could be the probable answers. Uh, Either both assertion and reason are true and the reason is the correct explanation. The second option would be the assertion and reason are true, but the reason is not explaining the assertion. The third is the assertion is right, whereas the reason is wrong. Fourth is the assertion itself is wrong, whereas the reason is right. So for all the assertion and reasoning question that will be following from now on, these will be the options. You can just type in the chat box as in A true or false or A and R are true, something like that. You can type. Can I wait for a couple of more minutes for you to answer this question? Is there any difficulty regarding this question? If there is some doubts, please let me know. Okay, so we'll just see the answer for this question. So before that, activated sludge, in the sense, there is one waste, generally it is a wastewater treatment process called activated sludge process. So in the that is a aerobic treatment process. So we have some microorganisms that can feed over the organic wastes in generally wastewater, let's assume municipal wastewater. So the microorganisms can feed over the organic content in the municipal wastewater, and as a result the microorganism is going to grow and multiply. Okay, right. As told, activated sludge is a process that we use for treating wastewater. So it is kind of a acclimatization that we have done to the bacteria. So before starting a new activated sludge process, we initially take some microorganisms and make them acclimatized to the conditions that in which we will be growing them, like to the waste, to the temperature of the waste, to the pH of the waste, and to all of this. So the assertion states that the activated sludge, so activated sludge is essentially going to contain the microorganisms that are growing in the activated sludge process. So the activated sludge will show higher cultivability compared to other environmental samples. First is just a true or false kind of a thing. It is true. Why it is showing higher cultivability? Because activated sludge contains organisms that are already adapted to control growth conditions. Like I told you, we will take some random microorganisms. Initially, before starting a full scale activated sludge process, we will make the microorganisms acclimatized to certain pH, temperature, and nutrient conditions. Like to the wastewater, let's say, we'll straight away we will not start the process. First we will give some time for it to adapt to the condition. So in that case, what we are doing, we are making the organism get adapted, which it is a kind of an enrichment process only. Whichever microorganisms are able to adapt to the conditions that we are supplying and grow, it will grow, and others will automatically be left behind over time. So in that case, as you have told, assertion and the reason is right, and the reason is the correct explanation for the assertion. So you have told that the controlled environment promotes a high density and activity of microorganisms. Exactly, because we are constantly supplying the same environmental conditions, the microorganism is able to show a good microbial growth and activity. The growth rate and the activity rate are high, because of which we get a high density of microorganisms. So the answer is both A and R are true, and R explains A. Okay. So the next question is, what is spatial heterogeneity and how is it important in environmental biotechnology? So first of all, you just give me what is spatial heterogeneity alone. Okay, so spatial heterogeneity ideally influences ecosystem stability, resilience, and functioning. Um, Before that, there are two kinds of heterogeneous nature that we can observe in the ecosystem. Like it can either be spatial heterogeneity or it can be temporal heterogeneity. So all this multi-organismic processes that we are uh, seeing in the nature is due to the spatial heterogeneity or temporal heterogeneity. Spatial heterogeneity means in a geographic location that we have, the distribution of conditions as well as nutrients is not going to be similar. It's going to be in a random distribution, and it's going to be different from one particular spot in that geographical area to another. So because of this difference in the nutrients and conditions in the particular geographic area, uh, there is going to be difference in microorganisms that are present there. So depending on the resources and the microorganisms ability to utilize the resource, the diversity of the microorganism is going to vary. Similarly, time is also an important factor which causes temporal heterogeneity. So, over time, how the conditions are varying and what kind of effects that has on the microbial dynamics, both of these are very important. Uh, in, in case of spatial heterogeneity, it creates microhabitats. So, within the given geographic location with specific microbial communities. So let's say I need to do an analysis in a particular location. If I'm going to only take a sample from one spot in that location and then find out what organisms are present, that may not be completely representing the entire microbial community in that location. I need to take samples from some 2 3 points in the same location and then see if there is any difference. If there is some difference, then we can find out, okay, what is the reason, like what kind of either physical conditions or nutrients, whatever is varying. We can try to see what is the reason and then correlate both the presence of the microorganisms with its metabolic function, and then have a holistic understanding of how the ecosystem functions. So that is the concept of spatial heterogeneity. And next question is another important question that forms the core of the first lecture in week 6. This I'll directly explain. So, what is great plate count anomaly? So, great plate count anomaly refers to the discrepancy between direct microscopic counts and viable plate count. So, I am taking some sample from a particular point. The same sample I am examining directly under the microscope, and I am also trying to culture the microorganisms that are present in the sample. When I'm going to compare both the results, ideally we expect that both are going to have the same count of microorganisms, but that will not be the case. There are various reasons why this is happening, but ideally, if I am going to have the same sample and a part of the sample, I am just going to view it under a microscope, and a part of it I'm going to uh, do culturing and then count the number of microorganisms. The numbers are going to be different. So this is what we also saw in one of the statements in the first question when we talked about this DAPI staining, right? So ideally, the microscopic counts exceed viable counts by several orders of magnitude. Some 2 to 3 orders of magnitude variations can be there in the viable cell counts. Okay, so this highlights a large gap between the number of microorganisms observed microscopically and those grown on laboratory media. This what does it signifies is that it reveals that the environmental microbes mostly cannot be cultured using standard techniques. Therefore, we can't use traditional methods which will make us underestimate the microbial diversity present in the uh, particular ecosystem. So that is why I gave the first question to give a general overview about the terms that we'll be discussing further. So we all know that environmental samples are going to be containing multiple microorganisms, and each microorganism has its own way of growing. It needs different physical and chemical factors to grow. So when we are going to do a direct microscopic count, either live or dead, based on the staining and all, we are just going to see the cell. We are just going to see and count, okay, this many number of microorganisms are present. But in the case of viable plate count, only the microorganisms that are able to grow and be viable or active are going to show their presence in the plates. Remaining, we are not going to account for them. So in that case, the number of microorganisms that we count using microscopic technique is going to be more than the number of microorganisms that are counted using plating technique. So this paradox is the plate count anomaly. Uh, I hope the concept is clear. If it is not, please let me know in the chat box. And in the meantime, I'll just post the next question. So the next question is kind of numerical answer question only. And the first part of the question we have already discussed in the first question. In most environmental samples, how much of total bacteria can be cultivated and how much of the bacterial phyla lack cultivable representatives. Just give numbers. Okay, so the right answer for this is around 0.01 to 1% of the total bacteria are only cultivable. 1 to 10% is a very high count. Um, so now you can just compare what knowledge of microorganisms we have and how much of it is still unexplored and unknown. And 70% of bacterial phyla lack cultivable representatives. So the three main approaches in microbial ecology as in kind of experiments or observations that we can do. So this question generally focuses on uh, the kind of real time or laboratory observations that we do. Based on that, what are the three main approaches? Two of the approaches, as I had already mentioned, can be real time or some kind of simulated environment, and there is one more approach. Okay, so culture dependent, culture independent, and bioinformatics and computational. Similar kind. Uh, Culture dependent is laboratory experiments. Culture independent in the sense in situ field. Culture independent can also be done in the laboratory. So, like kind of metagenomics or other meta analysis that we can try to do. Apart from that, culture dependent methods are in situ field observation, just monitoring the microorganisms, presence, dynamics in their local environment itself, instead of simulating the conditions in the laboratory. And the third one, the use of bioinformatics and computational approaches, we mainly use them for mathematical modeling approaches. So we try to um, get information from some ecosystem, and based on the ecosystem we try to frame a model so that in future we can theoretically estimate the microbial dynamics using the same model. So these three are the main approaches in microbial ecology. So the next question is, the microbial activities in any environment depend on three key parameters. What are the three parameters? That is the question. And these parameters are related to the microorganism itself. Okay, so almost right answers. Um, Microbial activities in any environment depend on the function or the type of species present. Uh, Diversity can be one way of expressing the functional ability of the microbial community, and their population size. Although the microbes are capable, only the number of microbes is going to have significant effect on all the functions that they are going to perform. And finally their physiological state. So physiological state, mainly the metabolic potential of the microorganism, how efficiently it can use the resources and what kind of products it is going to produce in order to allow the existence of other microbial communities. So, based on microbial cultivability, or how well we can culture microorganisms, we can classify the organisms into six categories. So, you can try to give some of those. Not necessarily you have to give all the six. How much ever out of the six you know, you can just post. So based on microbial cultivability, the microbes can be cultured microbes, which can be grown in the lab and maintained uh, as stock. The second category includes uncultured microbes, which are not yet grown, because potentially they are not cultivable, or we have not found enough techniques to uh, grow them in the lab. So either there are cultured and uncultured microbes. And next there is a category called culturable microbes. So these microbes, we know the conditions, and if we are using the right conditions, they can grow. Uh, it's not that easy as growing the organisms that come under the cultured category is, but still these are grown in the laboratory under right conditions. So that is the difference between cultured microbes and culturable microbes. And the next category includes non-culturable dormant microbes. So these microbes generally are present in a inactive state, and hence that becomes the major limitation for culturing these microbes. So these are non-culturable because they are dormant. And the next type is the non-culturable viable microbes. Uh, these microbes are active and viable, but still we are not able to culture them. There are other growth criteria that prevent uh, their culturing in the labs. And the last type is the non-viable or dead cells. So these are only present in their natural environment, but when we try to culture them, they become non-viable or dead. So that is why we are not able to culture them. So the six categories are cultured, uncultured, culturable, non-culturable, dormant, non-culturable, viable, and non-viable microorganisms. So if you understand what is the meaning of each of this six categories of microbes based on their cultivability, you can now correlate it with the fact that why only 0.01 to 1% of bacteria are cultured so far, and around 70% of the phyla, why they do not have um, representative microorganisms that we have already studied. And Ashwini, it's perfectly fine. You might have misinterpreted that you have posted the answer. It's completely fine. Now we'll just go with the next question. We have already seen about what is spatial heterogeneity. So spatial heterogeneity in environmental sample leads to what kind of organization? What level of organization? There is a specific term, but if you can post some phenomenon that is synonymous to the word that I will have in the next slide, it's perfectly fine. So spatial heterogeneity in environmental sample leads to a nested level of organization. So generally when we talk about spatial heterogeneity, in terms of microbial ecology, um, we can find a nested organization. Nested in the sense the microbial communities are not randomly distributed in the particular habitat. We can see some kind of patterns that are developed based on the resources that are available. So this pattern can be from different scales, like from a few centimeters to a few kilometers. We can identify different patterns. So through this we can decipher that the diverse complex communities are present in different population sizes across different regions in the same habitat. And this gives us the information about interconnected microhabitat. Uh, so the activity of one group, so because it is nested, the activity of one group that is going to be present at one part is going to affect the activity of another set of microorganisms that are going to be present nearby. So there is going to be a chain reaction. If one of the habitat is undergoing any significant change, it is going to affect the entire scale of microorganisms present from few centimeters to few kilometers. So that is why spatial heterogeneity gives us a nested level of organization. So the next question is, which approach in microbial ecology aims to study natural processes with minimal disturbance? So we have already talked about three approaches. One is the field observation approach, second is the laboratory experimental approach, and third is the modeling approach. Out of these three approaches, which of the approach aims to study the processes with minimal disturbance? Okay, so out of those three approaches, in situ field observation aims to study the natural processes with minimal disturbances. Okay, so the next question is a match the following question. Uh, the options are you have to match in situ field observation, laboratory experiments, mathematical modeling, and community function analysis with what they are deciphering. Ashwini, do you want me to repeat any of these questions? Okay, okay. I'll give you time for you to answer the match the following. Exactly, 3142. So, in situ field observation provides minimal disturbance to natural processes, and laboratory experiments they deliberately modify the conditions to reveal functional relationships or to understand the mechanism in which the microorganisms function. And mathematical models are used to generate theoretical knowledge based on the interaction studies. And finally, the community function analysis gives us information on what the microorganisms are doing in real time. Great. So one more interesting topic in this is the Vinogradsky chemoautotrophic uh, topic. So Vinogradsky is a scientist, and he had uh, studied, or after him, his invention is named as the Vinogradsky column, which we generally use to isolate or differentiate microorganisms. So what we do is we take a closed column which has a cover, and in that column we will supplement it with carbon and the sulfur sources. So once we cover it, the top layer will have some air. So aerobic microorganisms will be present at the top, followed by microaerophiles, because the concentration of the air is going to decrease from top to bottom. So microaerophiles will be present, and next the anaerobic microorganisms. So in the last part, we would have all this green sulfur, purple sulfur bacteria, and sulfate reducing heterotrophs are going to be present. So the whole idea is you just have a column and supplement it with some carbon and sulfur, and you will still find autotrophic organisms, but they are not going to use light as their source of energy. They are not photoautotrophs, but chemoautotrophs. So instead of light, you're going to supplement them with sulfur, and based on that you can identify and study the structure and function of those microorganisms that are present. So this is the concept of Vinogradsky column. Coming to the next question, it is again an assertion and reasoning question where the assertion is pure culture isolation is essential for understanding interspecies interactions, and the reason is pure cultures allow the study of complex community dynamics. You just have to tell if both the statements are right or if any of it is wrong. If you want me to tell the answer for this question, please let me know in the chat box so that I can continue. Okay, neither the assertion is fully correct nor reasoning is correct specifically. So yes, as you told, it is not fully correct, but we are not telling pure culture isolation is the only essential technique for understanding interspecies interaction. But yes, pure culture isolation is also essential for understanding interspecies interactions. Why? Because when we are going to isolate the organism as a pure culture and study it, we are going to understand the complete metabolic profile of the organism, and then finally using all this modeling approaches or all the computational techniques that we have, we can build a network between uh, all the microorganisms that are present. Let's say we have A, B, C, and three different microorganisms present in our ecosystem. We are going to isolate all of them separately as pure culture and going to do the structural and functional analysis and classification of these organisms, and going to have a complete understanding of the metabolism of each of the organism. Finally, we can make network analysis and then compare it with the real-time products that are observed in the real-time ecosystem. The reason is completely wrong because pure culture does not allow the study of complex community dynamics. Pure culture does not directly allow. As I told you, in assertion, it is a part, whereas in the reason, with pure culture, we can only study about one organisms, we cannot study about the complex community dynamics of the system. So the next question is to identify the true statements among the following. Statement 1 is all organisms lies equally. Lies in the sense all organisms break down or shed their cell wall equally during DNA extraction procedures. The next statement is enrichment allows dominant organisms to outcompete others under specific conditions. The third statement is that functional gene analysis can predict metabolic capabilities of a community. And the fourth statement is 16S rRNA sequencing allows precise species level identification for all bacteria. You just have to identify which of these four statements are true. Okay, so only C is true. Let's see. Both B and C are true. So the first statement is all organisms lies equally during DNA extraction procedure. That is false. Uh, Depending on the cell wall composition of the organism, some organisms can also resist the lysis. Uh, and this is an important reason that leads to extraction bias in the microbial study. The second statement is enrichment allows dominant microorganisms to outcompete others under specific conditions. So the rapid enrichment culture means we are going to provide some specific condition or a nutrient that will support the growth of one kind of microorganism over other. So it essentially means that the enrichment allows the dominant microorganisms which are capable of taking the help of the enrichment to outcompete the others in the specific conditions. So let's say we have two bacteria, one can grow in the presence of ammonia, whereas other cannot. While culturing, if we are going to supply ammonia, it means we are providing a enrichment media for the growth of these two microorganisms. So the organism which can grow over ammonia is the dominant organism here, and it is going to outcompete the growth of the other microorganisms which cannot grow with ammonia in the substrate. So that is why statement B is true. And the third statement is functional gene analysis can predict metabolic capabilities of a community because functional genes are the genes that are going to be transcribed into RNA and finally that are going to be translated as proteins. So these proteins are going to be the functional cores that drive all the metabolic processes, and hence when we are going to study about the functional genes rather than the whole gene analysis, uh, we can easily predict the metabolic capability of the community. And finally 16S rRNA sequence allow precise species level identification. The word species is what is leading to the statement being termed as false because 16S sequence can give us only details on the genus level classification and not species level classification. C is right, but B, on the other hand, is also right. I hope you have understood why B is also a true statement. If not, please let me know in the chat box or else try answering the next question. So in continuation to what we had discussed, the assertion is adding ammonium to a nitrogen-free medium will enhance the isolation of nitrogen fixers. The reason is ammonium supports the growth of nitrogen fixing bacteria. Okay, so assertion is false and the reason is also false. It is not true. Ammonium suppresses enrichment of nitrogen fixers because bacteria do not need, they can, if the ammonium is already present, they will not go for fixing the atmospheric nitrogen. So in that case, ammonium does not support the growth of nitrogen fixing bacteria, because if ammonium is already present, they are not going to fix the atmospheric nitrogen. And hen, henceforth, adding ammonium to a nitrogen free medium will not enhance the isolation of nitrogen fixers. We need free nitrogen in the medium to enhance the isolation of nitrogen fixing bacteria. So next question is about the CLPP technique. CLPP is used to investigate metabolic diversity at community level. It assesses substrate utilization profiles of environmental samples. So CLPP stands for community level physiological profiling. Okay, sure. Um, Somewhere in the end, we also have one more question about CLPP that we'll skip. I'll just give a overview. So CLPP is community level physiological profiling. So we are going to profile the microorganisms based on their physiology and in the community level, like uh, the microbial communities we are going to profile. So what happens is we take a microtiter plate, let's say, like we have also studied about microarray in one of the previous classes, no? So like that, we are going to have a microarray kind of a setup in which we will have different substrates, and we are going to add the same microbial culture to all the substrates that we are going to take. So if you're going to have the same culture, then all the diversity of the microorganism, everything is going to be the same, but their response to each of the substrate that we are going to add is going to be very different. And generally, in a multi-well plate, we are going to add all the substrate, all this, and finally we are going to quantify how much the microbial growth is happening. So this can either be via colorimetric or fluorometric methods, uh, depending on some UV absorbance or color or some fluorescence absorbance. We can estimate the microbial growth. This gives us this gives us the data on what kind of microorganism is present in the uh, primary inoculum that we added into each of this substrate. So if we are adding some different carbon sources or different resources with some sulfur or other nutrients that are present in it, if the microorganism can grow in all of this, it doesn't mean the single species of microorganism has grown in all the plates. Uh, the initial inoculum that we had added is containing organisms that can feed on a specific carbon source or a resource or a nutrient with sulfur. It can feed on different substrates. So what essentially it tells us is not the capability of a single microorganism, but about the diversity, metabolic diversity of different microorganisms present in the initial culture that we had added. So CLPP is used to investigate microbial diversity at the community level. That is true, because when we are using different substrates, different microorganisms that can grow on these different substrates are going to be present. If we see growth on only one or two of these substrate out of some 10 substrates that we are using, then we can confirm that there is very little microbial metabolic diversity that is being observed. And the reason is we are using substrate utilization profile, or we are monitoring how the microorganisms can feed on different substrates. So that is the concept of CLPP, community level physiological profile. Imagine you have a box, you split the box into 10, and in 10 you are adding 10 different substrates and adding the same culture of or same sample of microorganisms in all the 10 wells. After some time you find that in one case you find 8 out of 10 plates having good growth, which means 8 different substrates are consumed well by the microorganisms that you have added. Whereas in the other case, only two substrates show good growth. In that case, we can see that the diversity is very less. We can find only very little microorganisms which feed on a specific type of substrate that we have added. So it uses substrate utilization profile to give us information on the metabolic diversity at the community level. So the next question is, what can lead to the dominance of fast growing but ecologically less important species? So we talked about some kind of bias in one of the previous question. What kind of bias can lead to the dominance of fast growing but ecologically less important species? We talked about either extraction bias or enrichment bias. Out of these two, which can lead to the dominance of fast growing but ecologically less important species? So the options would be So the options for this question would be either extraction bias or enrichment bias. Which of the two bias can lead to the dominance of fast growing but ecologically less important species? Okay, so the answer for this is enrichment bias. So extraction bias, in the sense when we talk about this DNA extraction or about the cell lysis, we talked about the extraction bias. We are using some technique to break the cell open and then extract the DNA. For some microorganisms, the cell wall is easily breakable and you can easily take up the DNA. For others, it is not possible that way. So that is called extraction bias. Enrichment bias, in the sense like we talked in the case of adding ammonia uh, for enhancing nitrogen fixers. Ammonium cannot enhance the growth of nitrogen fixers, right? So in that case, if we are going to have a nitrogen fixer and going to add ammonium, that particular species is not going to grow, whereas some other species that is present is going to grow well in that condition. But it doesn't mean that we don't have nitrogen fixers in the sample that we are analyzing. This leads to a bias where we are depromoting the growth of the nitrogen fixer and promoting the growth of some other microorganism. So that is called enrichment bias, and it leads to domination of a fast growing but ecologically less important species. So this question also we'll just discuss the answer. The method that uses enzyme activity tests like catalase or oxidase is called biochemical characterization method. So we are using biochemistry um, and characterizing the enzyme. So it is called a biochemical characterization method. So next is kind of choose the right option. What is most likely to occur if only a high concentration of carbon source is provided during enrichment? So we are performing a enrichment isolation method, and in that we are supplying a high concentration of carbon source. Exactly. Heterotrophs will dominate because abundant carbon supports heterotrophic population enrichment. The next question is enrichment bias primarily results from. Right answer. Fast growers can outcompete the other organisms in lab condition. And next one is about the NCBI database. What is the use of NCBI database? Okay, so NCBI stands for National Center for Biotechnology Information, and we have a provision for BLAST, basic local alignment search tool, through which we can find the homologous sequences. So it is primarily used for sequence similarity search. Okay, so we talked about the CLPP previously, so I'm skipping this now. So the next question is isolation using agar plates allows for selection of pure colonies, and agar medium restricts all microbial growth except the target organism. Are both these statements right? Okay, right. Assertion is true and the reason is false. So agar plate generally make allows for selection of pure colonies, because if you are going to do proper cereal dilution and plate on it, different microorganisms can grow, but each of it will grow into a particular colony, and we can identify those colonies, and we can isolate pure cultures from that. Whereas it the statement that says that agar medium restricts the microbial growth of other organisms except target organism is false, because agar is generally a solidifying agent, and it does not have any role in allowing for selection of microbial growth. The answer is perfectly right. So next, I actually forgot to add a question slide before this, so we'll just discuss. The assertion is MPN test can estimate counts for metabolically specialized group of microorganisms like iron reducing organisms. The use of selective media enables targeting specific metabolism. So here that both the assertion and reason are right, and reason explains the assertion. Selective enrichment allows assessment of specialized microbial counts. Okay, so the next question is to identify the false statements. Okay, so here B and D both are false. The purity of an isolated culture can be assessed via microscopic and colony characteristic tests that we have already seen. MPN test does not rely on visible colony count. It relies on a statistical assumption of the number of microorganisms, or the statistical analysis of colony count. It does not directly rely on visible colony count. So that is why B is also a false statement. And in case of C, taxonomic affiliation requires phenotypic and genetic comparison with known strains, which are called as reference strains. And the fourth statement is the naming of new bacterial species can be validated solely based on gene sequence similarity. No, we also have other phenotypic factors like mentioned uh, in the naming of new bacterial species. So both B and D are false. B is false because MPN is kind of a statistical technique. It uses some statistical approach to give us the microbial count. Okay, so the next question, uh, when there is a culture that consists of only one microbial species confirmed by both microscopic and growth test, what do we call the culture? The answer is not a pure culture. So ideally, we would start thinking about pure culture. It is axenic culture. Dash is essential for the isolation of anaerobic microorganisms due to its restricted oxygen environment. So here the question is about what kind of technique we can use, what method we can use. We just have few more questions left, and if you are comfortable, you please let me know so that I can just continuously go through all the questions and answers instead of making you answer the question. Anything is fine with me. Can we just discuss the question and answer slide by slide? Okay, so we need a roll tube method. So generally to isolate anaerobic microorganisms, we use a technique called roll tube method. So can I go through the next questions one by one without waiting for you to answer? Is it fine with you? Okay. So the next question is, what feature best validates an axenic microbial culture? So we talked about axenic culture a couple of slides ago. Um, any culture can grow on a agar plate. It doesn't mean like only axenic culture can grow. Uniform staining and morphology. So what we saw about axenic culture, it is a culture in which only one type of microorganism is present, and we have confirmed it through microscopic and culturing techniques. So when we are going to confirm it through microscopic techniques, we are going to use some staining method. If the culture is going to contain only one type of microorganism, there is going to be uniform staining and uniform morphology. So that is going to be the answer for this question. Any microorganisms can grow on a agar plate. Metabolic activity will be there. Antibiotic resistance may or may not be there, but the factor, as you mentioned rightly, is uniform staining and morphology. The next question is, how is axenic culture confirmed and why is it critical in microbial ecological studies? So, axenic culture is ensured by demonstrating uniform traits in terms of structure and function. So, first of all, we do some microscopic analysis. We see how the microorganism is looking. If there are different types of microorganisms, then they can respond differently to staining, and also the morphological features, the cell wall thickness or the presence of other appendages or some flagella, everything is going to vary. So if all of it is uniform, then we can confirm through initial microscopic analysis. And next is the colony morphology across subcultures. So we are going to grow the organism for another 2 to 3 generations. We are going to subculture it sequentially, and then even after subculturing, if the morphology is same, then we can confirm, okay, we have had a pure culture and we have been culturing it, and it is going to show a consistent growth in different medium. Like if I'm going to have a carbon rich medium or a sulfur rich medium, the growth is not going to be same in both the medium, but it is going to be uniform in one of the given medium. If it is going to grow rapidly in carbon substituted or carbon rich media, then the entire culture is going to show a rapid growth. In sulfur, if it is not going to grow, then there is going to be zero growth in sulfur. Then it means we have a pure culture, and either all the organisms can grow or all of it is die. And the next thing is, confirming an axenic culture is crucial because mixed cultures may give misleading results regarding microbial activity and taxonomy. uh, we can see rapid evolution in microorganisms. Therefore, getting false positive results is very, very easy. So, we have to have multiple tests to confirm that we have only a axenic culture. So, the next question is about the most probable number technique. So, the most probable number technique is used to estimate the viable microbial population in samples, in diluted samples. So, if you have a a concentrated sample, you have to dilute the sample. If you already have a diluted sample, then MPN is one of the best techniques to count the number of microorganisms present in that sample. And one more catch here is it is going to give us only the number of viable cells, the cells that are actively growing. Only those cells we can count. So what we do is we take a broth and we inoculate it with microorganisms like given in this slide. uh, we are going to inoculate different concentrations in different concentrations. So, and in each concentration you will get some kind of results. When you are going to add uh, 10 ml or 1 ml of the broth or zero point 0.1 ml of the broth, you are going to have different results that you can observe. So there will be one table, like we have studied about this t test, p test in statistics, like that there will be one table. So let's say the table will have different combinations. So in each of the condition you will have some 5 tubes. So like 555 or 554, 553, like that there will be different combinations of numbers. So in 10 how many tubes are growing, in 1 how many tubes are growing, and in point 1 how many tubes are growing. These three numbers together will form a triplet uh, series, right? With this series you are going to compare it with that standard table that is present in the MPN test, and you will find the number of organisms that can be present. So that is why we call it as a statistical based method rather than direct counting of microorganisms. So based on the combination that we are going to observe here, so here what is the number 4, 2 and 0. So a table will be there in which there will be 10, 1 and point 0.1, and here there will be number of cells that are estimated. So in 4 to 0 combination, if there are going to be 50 count, let's say for example, then it means that in your sample 50 organisms were present. So, and after this, this is the presumptive test, and after that you go for a confirmatory test in some specialized broth, and you are going to monitor the growth. And after that, you are going to confirm the results that you got in confirmed test using a completed test. So, this is the complete procedure of MPN technique. So the next question is, fermentative organism enrichment requires only electron donors without external electron acceptors, and fermentation uses endogenous electron acceptors generated during glycolysis. Here both the reason and assertion are right. So the what are we telling? We have some fermentative organisms, and if we are going to enrich, if we want to enrich them, we have to supplement some electron donors, and not we are not going to supply any external electron acceptor. So why are we doing so? Because in fermentation process, endogenous electron acceptors are generated during the glycolysis process. So only when we give donors, these acceptors will have donors for electrons, and the fermentation process will continue. So the next question is, chemolithotropic bacteria can be enriched under aerobic and anaerobic conditions, and the reason why we say so is different electron acceptors can substitute oxygen in anaerobic chemolithotropic process. True. When we talked about redox tower, we discussed this. When oxygen is present, this chemolithoautotrope first uses oxygen. After that, it goes for other electron acceptors like nitrate, sulfate, and iron. So in that case, chemolithoautotropes can grow in both aerobic and anaerobic condition. First, it will consume all the oxygen, and after that, it will go for other acceptors like nitrate, sulfate, and iron. Okay, so the next 2 to 3 questions will be of the same type. So we will directly see the answer. When we are going to go for some direct isolation, we are going to have individual colonies obtained directly. When we are going to go for some enrichment based method, we are going to use some specific substrate for enriching the growth of only one type of microorganism. If we want to isolate pure culture, It means that we want a single organism without contamination, kind of a axonic culture. And if we want mixed culture enrichment, then we have to supply substrate which contains the growth factors required for the growth of different microorganisms. So that means multi-species assemblage growth. Multiple species are going to present, and all together in assembly we are going to grow them. And this too we have already discussed in previous 2 to 3 classes also. 16s rRNA in prokaryotes you can find 16s rRNA, whereas in eukaryotes, so eukaryotes the ribosome is going to be different. It's 80s ribosome, whereas in prokaryotes it's 70s ribosome. So in bacteria and archaea 16s rRNA gene we are going to use, whereas in fungi and algae 18s rRNA we are going to use. The metabolic characterization we are going to do for all isolated pure cultures. So when we have pure culture, we will be able to identify what is the metabolism or metabolic functions of the particular uh, strain. And in case of ecophysiological studies, environmental stress tolerance is the basis of ecophysiological studies. How ecology affects the physiology. So there comes this environmental stress tolerance. Next one is about the enrichment conditions. So we'll start from option D. So high temperature, temperature in the sense we are talking about thermophiles. And the next EC option is, if you are going to talk about aerobic organisms, then they're going to be lithoautotropic. I mean lithotropic, aerobic, and ammonia we are going to provide. So it is chemolithotropes. Anoxic condition, there is no oxygen, then we are going to go for fermentation if there is no oxygen. And if there is oxygen, then we are going for phototrophic, because oxygen plus light, it can harvest energy from light and be phototrophic with the use of inorganic carbon substrate called carbon dioxide. So the next question is, why might some organisms from enrichment cultures remain uncultivable as pure culture? Um, because they require interspecies interaction. Like when we discussed about anaerobic digestion, we saw that first hydrolysis should happen, and then fermentation will happen, and then methanogenesis will happen. If the fermentation itself is not happening, then the methanogens cannot survive there. They don't have any substrate to consume and produce methane. So that is why some organisms in enrichment culture remain uncultivable as pure culture, because they require the presence of other microorganisms which help them to grow. So this we have discussed enough now. What the difference between direct isolation and enrichment isolation? So this question I will upload the slides in the drive, and then you can uh, look for the explanation. So thank you for attending today's session, and if you have any doubt you can post in the chat box. Or Ashwini, I really appreciate your participation in the class. Thank you so much. Okay, okay then. I'll close the session now. Thank you so much.